Hello, my name is Martin Christensen. I'm a Danish medical student at Hans Christian Andersen Children's Hospital in Odense, Denmark. My study is called Hypothermia for acute insulin-induced brain damage, a rat study, and this is only the pilot study. So, neurodevelopmental impairment affects up to 50% of children born with CHI. They have symptoms as learning difficulties, cortical blindness, memory problems, motor delays, and more. These complications arise due to hyperinsulinic hypoglycemia events. We know that neurons rely on glucose as their primary fuel and ketones as their secondary. In CHI, insulin secretion is unregulated and thereby making the blood sugar low, while also inhibiting the ketogenesis. We know that the neonate brain has a much higher need of glucose than the adults, making it more vulnerable. So with no glucose and no ketones, the brain cells will die and irreversible damage will occur. So Henrik and I asked the question if there is something to do in these situations with severe low blood glucose due to these episodes. And we know that hypoxia shares some similarities with hypoglycemia. In both these conditions, the brain is deprived of energy. For hypoxia, the children is treated with therapeutic hypothermia, and that's a well-established treatment for hypoxic ischemic encephalopathy. The treatment protocol for therapeutic hypothermia is that we're cooling the child's body temperature down to 33.5 degrees Celsius for 72 hours. And by that we slow the metabolism and protects the brain. So our hypothesis is that cooling may also prevent brain damage shortly after severe hypoglycemia and congenital hyperinsulinism. So I did the pilot study. I had a litter of 14 rat pups by the age of 14 days. At 14 days old, their brain's development is comparable to that of a firm-born human infant. I split the groups into a fasting group and a non-fasting group. The fasting group fasted for 6 hours, and the non-fasting group did not. One rat in each group served as controls. So the intervention groups got an injection of 10 units per kilogram fast-acting insulin intraperitoneal, and every 30 minutes I monitored their blood glucose levels by the tail vein. 4 hours after, they all got a rescue with 50% glucose and then reunited with their mothers. 20 hours later, the rats were euthanized and perfused with 4% performaldehyde As you see here, perfusion is very important. If you're not making a well-perfused brain, there will still be some blood left, as you see on the first year. And on the second, it's a more successfully perfusion. And we do that to fixate the tissue. After the perfusion, the brains were stored for 6 hours in performaldehyde and then replaced to containers with 20% sucrose for 48 hours. After that, they were fully saturated and ready for freezing and cutting. To quantify the brain damage, I used fluoride B. It's a staining procedure. The fluorescence in the staining binds to brain cells undergoing apoptosis or necrosis, dying cells. So this is the result of the glucose monitoring experiment. As here, there's a non-fasting group and a fasting group. The fasting group started out with a little lower blood glucose level at 5 to 6 millimoles per liter and reached 1 or below 1 for 2 to 2.5 hours before they got the rescue. And the non-fasting had a little higher blood glucose level at the beginning at 7 to 8 and could only maintain a blood glucose level at or under 1 millimole per liter for 1 to 1.5 hours. So yeah, there may be a little bit too much light in this room, but you see some green dots over here. You can't really see all the tissue, but the green dots, that's the fluoride B positive neurons, the neurons undergoing cell death. For the fasting group, all of the rats except one had a lot of these neurons with a mean of 402 positive cells per section. For the non-fasting group, only 2 rats had detectable fluoride B positive cells, and these 2 rats had also a lower blood glucose level throughout the experiment, so it makes sense. But it's very obviously that to use this model, you need to fast them prior to insulin injection to be sure that you can detect brain damage by fluoride. So the perspectives on this study is that it makes the groundwork for my next study where I will test this hypothermia treatment out. I wish I had done it for today, but it is what it is. So the rats will undergo 4 hours of cooling after fasting and insulin injection. And then they will be stained, and hopefully I will see a less number of these positive fluoride B cells, indicating less brain damage. And ultimately, one day in the future, this can be a treatment for children undergoing these extreme hypoinsulinemic hypoglycemia events to prevent some of this brain damage. Thanks.